Okay, welcome everyone. We'll wait a few more moments for people to join, and then we'll start today's seminar. Okay, I think in the interest of time, we might get started now. And next slide, please. Perfect. Okay, welcome everyone today, and thank you for joining us for the seminar entitled Harnessing RNA Mobility for Effective Plant Protection, presented by Dr. Chris Bosnian. Next slide, please. I'm Narelle Manzi, and I'm the Scientific Manager for the ARC Hub for Sustainable Crop Protection based at Coffee within the Centre for Horticultural Science. We're located at the Queensland Bioscience Precinct on the beautiful St Lucia campus. And before we start today's seminar, next slide, please. I'd like to start by acknowledging the traditional owners and the custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Next slide, please. So those of you who are familiar with the format of these seminars, it will run from 12 noon until 1 p.m. And at the conclusion of the seminar, we'll hold a Q&A session. So if you have any questions that you'd like answered, and I encourage everyone to try and come up with a question, please use the Q&A button that you can see in the bottom center of your screen. Please don't use the chat button, just the Q&A button, and we'll address the questions at the conclusion of the seminar. Next slide, please. So today, I'm really honoured to have the opportunity to introduce the speaker today, which is Dr. Chris Bosnian. I've had the privilege of working with Chris for several years now, and I'm always inspired by his passion, his dedication and enthusiasm for understanding not only the fundamentals of RNA interference, but also how these can be harnessed to provide plants protection from pests and pathogens at research level and more recently at industrial applications. So a little background about Chris. His career began here at the University of Queensland, where he completed his PhD looking at graft transmissible movement of RNA interference under the guidance of Professor Bernie Carroll, who most of you would be aware of. From there, he took up an amazing opportunity to work at the prestigious ETH Zurich, from which there are many notable alumni, including Albert Einstein and numerous Nobel laureates. Including Chris will be among them one day. And while Chris was there, his research was supported by both an EMBO long-term and Marie Curie incoming postdoctoral fellowship. And these enabled him to investigate his cell type specific RNA-mediated gene regulation and movement. On his return to beautiful Australia here, obviously looking for some warmer weather, he worked briefly on the model nematode C. elegans. And then we were lucky enough to have him join our group here at Coffee and becoming part of the ARC Hub for Sustainable Crop Protection, where he's now investigating the movement and mechanisms of RNAi-mediated biopesticides. We are thrilled to have him as part of our group, and I'm really excited to have him present his work to you all, and I'm sure he'll be taking us on a wonderful tour of RNA interference and how it can be used to help save the world by producing more food for everyone. Thank you, Chris.
Thanks, Narelle. Can everyone hear me? I assume that's a yes. Thanks for the introduction. I don't know about this Nobel Prize talk, but it was very nice of you. I'd also like to begin by acknowledging the traditional owners and the custodianship of the lands on which we meet today, and pay respects to their ancestors and their descendants. So I'll apologize in advance for those of you who are not molecular biologists. This will be a very molecular biology heavy seminar, but that's the area which I work in. So I'm going to talk to you today about my work loosely titled Harnessing RNAi Mobility and Functionality for Enhanced Plant Protection. So that's slightly different to the title I've given outwardly, but this is really what I'm going to focus on today. So I'll just start by introducing the work I've done previously. So this kind of introduces why I've come to the field I'm in now and, sort of the expertise I've gained over the years and how this is really going to benefit crop protection and what I'm doing here within coffee. So my main field of research is in RNA-mediated gene regulation. And the two main focuses of this are microRNAs. So many of you will be familiar with microRNAs. They're conserved in all eukaryotic organisms. They're endogenously encoded. They're a non-coding RNA, which gives a large foldback structure to give a single processed microRNA. So this microRNA is very important. There's many of them in plants, mammals, all eukaryotes, as I said. And these are key to development and stress responses in all of these organisms. Another example of RNA-mediated gene regulation is RNA interference. And this is mostly what I'll be talking about today. So RNA interference is very similar. It differs in that it has a long double-stranded RNA precursor, and it gives multiple species of small RNA. Again, it's processed by dices, but it's more involved in plant protection against viruses, transposons, and maintains genome integrity as well. So these two kind of converge at the effector stage, and this is where they're loaded into Argonaut proteins. So Argonaut proteins form something called an RNA-induced silencing complex to downregulate genes. So this can happen at the RNA level, where RNA is cleaved and is turned over. It can be translational repression, or it can also actually be DNA methylation, chromatin compaction, and these type of things. That's how transposons get silenced. So the four major topics I've worked on during my career. One is cell type specific profiling of microRNAs. So I really wanted to get to the mechanisms of how microRNAs work at a cell type specific resolution. As Narelle mentioned, when I moved back to Australia, I transitioned to the model nematode C. elegans at the Queensland Brain Institute, and I translated a lot of my research from plants into C. elegans to answer similar type questions, which are not possible if you don't get cell type resolution. One of the overriding themes of my research over the years, from my PhD work all the way up until now, is the movement of both microRNAs and RNA interference. And this is something I'll really delve into when I talk about utilizing this for plant protection. And that leads me to the main part of what I'll be talking about today, and that is my dissection of the mechanisms and movement of RNAi-based biopesticides for crop protection. So this is the main part of the work I do within Nina's lab and within the ARC Hub for sustainable crop protection. So I'll just briefly go over some of my previous work because, it's topical to what some of the people in coffee do. And it sort of introduces why I came to where I am now. So the first part of this work was cell type specific microRNA loading, movement, and action in the Arabidopsis root. So I chose the Arabidopsis root because it has stereotyped cell files, which we can really pull apart to define the activity of each of our microRNAs in these cell types. The Arabidopsis root is a beautiful system because it has cell type specific promoters where we can define all of these cell types really, really beautifully. So we can define the columella, the stele, the endodermis, the cortex, and the epidermis. And what we can do with this is drive expression of one of the main effectors of RNA silencing or microRNA-mediated gene silencing, RNA interference. What we can do is pull down microRNAs or small RNAs from each of these individual cell types. So we can develop a map of how these microRNAs are expressed, loaded, and how they act in each of these cell types. At the same time, I wanted to look at the transcriptome, so the entire genome landscape of transcription in each of these cell types as well. And I used the same set of cell type specific promoters, but instead of driving an Argonaut protein, I decided to drive a component of the ribosome. So the ribosome is an actively translating mRNA machine, and I was able to pull down actively translating mRNAs from each of these cell types. I wanted to cross these two data sets together and get an idea of how microRNAs work at a cell type specific level. This is the type of information we got. Each cell type has a really unique profile of loaded microRNAs. Perhaps not surprisingly, but this is the first time this has actually been shown for Argonaut loading. We crossed this with the transcriptome profiling, so the translatome, which I came up with from ribosome profiling, and we could see there's unique mechanisms of microRNA-mediated gene regulation in each of these cell types. So we crossed all of this together, and we have an online browser which we can really delve into the mechanisms, and we found really unique mechanisms of microRNA-mediated gene regulation, which would have escaped every sort of analysis if we didn't look at cell type specific profiles. What we also saw from this was that there was widespread microRNA movement, which has previously escaped a lot of people's attention. So as I said, when I moved back to Australia, I wanted to transition to a different organism. I was always fascinated by C. elegans, and it has many similarities with the Arabidopsis root in that we can define individual cell types with specific promoters. So in this case, I defined the intestine, the muscle, or all of the neurons. So that's 300 cells, all neuron cells within C. elegans. And I did a very similar approach to what I did previously. So I tagged the Argonauts in C. elegans. There's 2 of these which bind microRNAs. And I did deep sequencing of these as well. So what we saw was different cell types have different profiles of loaded microRNAs. Within these individual things, we even have differential Argonaut loading. And within this, we have spatial and temporal flexibility of microRNA loading. So during aging, microRNAs can actually switch between Argonauts dependent on their expression patterns and the microRNA itself, what it does during an aging process. So perhaps the most relevant set of data which I previously generated to what I'm doing now involves movement. So movement of sRNAs and RNAi in particular. As Narelle mentioned, my early work with Bernie at SCMB involved looking at graft transmissible small RNA mediated sciencing. You can see an image of that here. And I also looked at cell-to-cell movement. So this is a sensor of cell-to-cell small RNA movement. So 10 to 15 cells out from the veins of the leaf. This is a monitor of movement of small RNAs. So there's multiple aspects we wanted to look at. The first was how do small RNAs move from cell to cell? This was an open question in the field when I entered it. And there was either 2 options. One, they could be moving between cells via a symplastic pathway. So this is the junction between 2 cells defined as the plasmidesmata. Or the small RNAs could exit the cells and travel through the intercellular space to rejoin the cell, the adjacent cell type. So by blocking the symplasm using different molecular tools, we were able to show unequivocally that small RNAs move cell to cell via the symplasm. So this is an example of blocking. We see a lack of this bleaching, which shows that the small RNAs are not moving cell to cell. We kind of combine this with my cell type specific small RNA sequencing to show that as small RNAs move across these cells, so adjacent cell types, they actually get loaded into their cognate Argonaut. And this is something we term consumption. So small RNAs move from cell 1, 2, 3, 4, and they're loaded, forming a sort of downward gradient as they're consumed. If these are not loaded, based on their sequence context, they in theory have an unlimited movement regime. So this is all this work which, kind of gave me a level of expertise in mechanisms of small RNAs and also movement. And I really wanted to apply this to the work that Nina was doing. And I joined the lab to look at Foliar dsRNA application for RNAi-based pest and pathogen control. So I wanted to look at how we could really optimize mechanistically this system. So we all know, based on Nina's work, that spray RNA, so we spray double-stranded RNA onto a plant. We utilize the plant's endogenous RNAi mechanism, or actually the pathogen we're going to attack. So this is processed via dices into small RNAs, and these small RNAs target via Argonauts, target transcripts within our pathogen of interest, or a virus, in fact. So what we know is that, early work by Nina showed that topical application of double-strand RNA can yield virus resistance. And this is an ongoing theme in the lab. Carl Robinson works on this continually, and it's really an effective system. And Sawyer and Beck Degnan within the lab, well, Beck's not directly in the lab, they showed that double-strand RNA sprays can actually be really effective at controlling, in this case, myrtle rust. So this is one example of fungal control. Ritesh who was in the lab and is now left to greener pastures, he actually showed that we could also use spray-mediated gene silencing to get resistance to, or at least protection against whiteflies, so insects. So we can see virus, fungal, and insect controls based on spray-mediated double-strand RNA applications. However, many of these do not reach this sort of mythical 80% efficacy which we look for. I wanted to sort of investigate how we could routinely meet this without, having unknowns. So I wanted to look at using one, RNA movement. So I wanted to look if we could spray RNA on a plant, if we could get whole plant protection. So if we could optimize this, could we increase the efficacy of every spray we use? And secondly, I really wanted to optimize the functional component of RNA interference. So I wanted to look at small RNA production, small RNA amplification, and if there's any way we could really predict and really enhance these to get more efficient RNA interference against whatever pathogen we chose to look at. So being a fundamental biologist by trade, I chose to start with the model plant Arabidopsis. If anyone doesn't know this plant, it's a common weed throughout Europe and the US, and it's the model plant for anyone working in plant molecular biology. So I started by adding double-stranded RNA. In this case, it was fluorescently labeled with a fluorophore, and I did sections of the leaves. So I wanted to know if we added double-stranded RNA to the leaf, could it enter the vasculature of the leaf? The answer is yes, and we can see this green signal here entering the vascular bundle in general, and we can also see that it enters the two main transport systems within the vasculature of a plant, namely the xylem and a little bit in the phloem. So this basically gives it the beginnings of something which could potentially move to distal tissues, the first step in the whole process. So I wanted to really see if double-stranded RNA was there. I didn't want to use a fluorophore, and I wanted to know if intact double-stranded RNA was there. So I stepped back in techniques many years and used northern blots to address this. So northern blots are really the only way, apart from maybe a nanopore sequencing technology, to look at full-length double-stranded RNA to see if it's present. So I used a mechanical separation technique, again adding double-strand RNA to the leaves of the plant. I mechanically separated the vasculature, and what we could see was a full-length double-strand RNA within this. And we know this is likely within both the phloem and the xylem based on the fluorophore I showed previously. So next I looked at distal tissues, and we could see efficient movement of double-strand RNA to the aerial part, including the apex, the flowers, the stem, and even the seed pods. So we could see potential transmission to seeds. Looking at the below ground parts of the plant, we saw really nice and rapid movement of double-strand RNA as an intact molecule to the roots, so anywhere from 1 hour up until 24 hours. So obviously Arabidopsis is, not really a relevant crop species. So I wanted to translate out of this to see if double-strand RNA could move upon applications. So looking at the Solanaceae, I looked at tomato first, and tomato was a really good system for this. We saw nice movement to the reproductive tissue, really, really efficient movement to the new leaves, so young leaves pre-formed at the time I added double-strand RNA, and a little bit of movement to the roots as well. Switching to another Brassicaceae, so it's sort of relative of Arabidopsis, but way more commercially relevant. I used canola, and canola again, while slightly different, had nice movement to the stems and flowers, depending on which floral organ we looked at. It also had some movement to the roots. The movement to young leaves was not so efficient. So each species is slightly different in that if you compare tomato and canola, they are slightly different. And stealing something from Ritesh's work, he actually showed that if we spray double-stranded RNA onto the leaves of cotton, it also moves down to older leaves. So this is an advantage because we can spray the top of a canopy and still get double-stranded RNA moving to the older leaves underneath that canopy. So as opposed to moving to the younger leaves, we can also see movement down the plant. This is work done by Safira, a PhD student who submitted recently, and we wanted to know, and other people have kind of shown this in the same similar way, we wanted to know if we add a double-strand RNA to hydroponic media, do we see it move to the younger leaves of a tomato plant in this case? The answer is yes. Within 3 days, we saw nice levels of double-strand RNA, I should say, intact within the leaves of tomato. So this is important for, applying this to protected cropping environments and this type of thing. So I wanted to know how long this is maintained. So The distinction between movement and maintenance is a bit hazy here, but I wanted to know if I had a double-stranded RNA, switching back to Arabidopsis here, and I waited 14 days until I had new growth. So this is a combination of movement of double-stranded RNA and then likely maintenance in this new tissue. Do I still see intact double-stranded RNA? And the answer is yes. So we see intact double-stranded RNA in new growth, which is likely moved there and then being maintained. So we see this up to 2 weeks post-application. So this is not an isolated occurrence. I'd like to thank Anne and Ivy for letting me share their little bits of results here. So it also works in avocado using trunk drilling and spray applications. It works in Metaceae, and Ivy has shown not the greatest system is sugarcane, but sugarcane does work if you give, potentially it works if you give high pressure spray applications. So how does double-strand RNA move? We know it moves, but I wanted to know what channels it moved through such that we could, optimize what pathogens we could test, what growth conditions we could test, whether it was nutrient stress, whether it still moved. So these are things we needed to address in terms of how it moves. So as I mentioned earlier, small RNAs move through the symplasm. So we showed this in previous work I did. So they actually are within cells. They move through this channel called the plasmidesmata onto the next cell type. So does long double-stranded RNA move in the same way? So if we spray it on the top of a leaf, it enters the vasculature. To get to the vasculature, is it actually traveling around cells, or is it entering cells and moving cell to cell in that way? So to do this, I use my sensor of small RNA movement that I introduced earlier. So this is, again, showing 10 to 15 cell movement from its expression domain. So we express an RNAi transgene from the veins of the leaf, and the small RNAs move 10 to 15 cells out to give this bleaching phenotype. So in our control plants, see these are untreated plants, we can see, yes, there is small RNA movement. When we block this using callose deposition, so this is just phosphate starvation, which actually induces the blockage of the symplastic channel, we see no small RNA movement in the younger leaves. So what I wanted to do was apply double-stranded RNA to these under these treatments with the control of the small RNA movement in the background and see if we saw any change in double-stranded RNA movement. So using Arabidopsis and one of our best systems, which was moving to the root, you can see under the control, small RNAs move very efficiently. So as we'd expect, we see the bleaching, and we see double-stranded RNA move to the apex and to the root of Arabidopsis. When we blocked the symplasm here, using either constitutive blocking or vascular blocking, so this was a complicated transgene I won't go into, while we blocked small RNA movement, we didn't block double-stranded RNA movement upon top of application. So it moved just as well to the apex and to the root under both of these treatments. I wanted to know if that maintained double-strand RNA pool after 14 days. So when we added and looked in new tissue, did we see this was also mobile under a symplasm blocking system? The answer is yes, we still saw double-strand RNA move when we blocked the symplasm. So this strongly implies that foliar applied double-stranded RNA is not moving via the symplastic channel. So it's quite different to the small RNA pathway. So the alternative, obviously, is it within the apoplast? So is double-stranded RNA moving via an apoplastic channel? The apoplast is typically involved in water transport and also for nutrient uptake and transport. However, there is also evidence showing that small RNAs are contained, or RNA and many RNAs actually are contained not only within the apoplast, but they can also be contained within vesicles within the apoplast called extracellular vesicles. So we wanted to address whether the double-strand RNA we apply is in one of these compartments. So by extracting the apoplastic fluid from either the mobile leaf, so the destination leaf in tomato, or in the roots of Arabidopsis, we saw a nice signal in both for a full-length double-strand RNA, showing that the double-strand RNA, while not moving symplastically, is actually present within the apoplast. On a side note, we extracted extracellular vesicles and we didn't see any signal here, implying that it's likely free within the apoplast and not contained within some form of vesicle. And we saw the same thing when we looked at the maintained dsRNA 2 weeks after application. So we saw a nice signal again here, implying that both mobile and maintained double-strand RNA are present within the apoplast. So when they move to these tissues via the apoplastic channel, I wanted to know, are they actually functional? So we know they're functional when we spray on a single leaf and look at viral infections, or with Ritesh's work or Anne's work with fungal and insects. I wanted to know, does double-strand RNA move to new tissues, and is it functional in these tissues? So to start with, I used, again, the Arabidopsis movement to the root, and we used fungal pathogens infecting the roots. So I used GFP-expressing verticillium and GFP-expressing fusarium. And what I could see was a subtle yet reproducible downregulation of GFP within these. So we added GFP double-stranded RNA to the leaf, it moved to the root, and it downregulated GFP within the fungus. So it does move to the root, transfers to the fungus, and is functional. I wanted to know, if we moved in the opposite direction, could we still get functionality? So in this case, we looked at double-stranded RNA moving, in this case, Nicotiana benthamiana, which is a model for virus infections. That's why we chose to move to this. Double-stranded RNA via a different application method moves to the newer tissues here. So you can see a northern blot showing that. When we infected with the virus, so again, we were just using GFP here, GFP double-stranded RNA moving to the new leaves, and infected with a virus expressing GFP, we could see downregulation of two different viruses, so GFP from TMV or GFP from PVX. So this shows that double-stranded RNA, when it moves to aerial parts of the plant, can also be functional. In this way, it was targeting viruses. So the key molecule within all of this are small RNAs. So far, there's been no evidence of where these small RNAs are produced. We know the double-stranded RNA moves to the root, but the small RNAs are somewhere within this whole system, actually knocking down GFP in our system here. So where are they actually produced? The first scenario is that they could be produced within the plant tissue. So this is an infected root with verticillium expressing GFP. The double-stranded RNA could move from the leaves to the roots and then be processed by the plant RNAi machinery, then transferred to the fungus. Or the other option is the double-stranded RNA itself within the apoplast transfers to the fungus first, where the fungal RNAi machinery first processes into small RNAs and next actually targets the fungal transcripts, in this case, GFP. So in order to address this, I use small RNA sequencing. It's a bit more complex than what I show here, but I'll purely focus on the GFP part here. So this is double-stranded RNA targeting GFP, and we can see small RNA peaks. So these are reads from small RNA sequencing against GFP. So this is in a wild-type Arabidopsis plant. So wild-type plant, double-stranded RNA infected with verticillium, and we sequence the small RNAs from here. So how do we distinguish whether it's happening in the plant, whether it's happening in the fungus? Arabidopsis, as I said, is a model for basically all forms of genetics in plants. So I already had a knockout of all of the dices which are responsible for processing this double-stranded RNA. So in this scenario, there is no possibility of this happening. And if we saw no small RNAs, we could say it's being processed in the plant. However, we saw a very nice signal of small RNA production in the absence of all plant dices capable of processing this double-stranded RNA. So this strongly implies that double-stranded RNA moves to and is processed in the fungus. I won't go into this, but this is a way of analyzing amplification of these small RNAs. So we were able to show that this is actually happening. The take-home message from this is, in my opinion, double-stranded RNA moves to distal tissues where it transfers to foreign pathogens and is processed by their RNAi machinery. So this is all the movement part, and this is something I'd really like to build into the whole RNAi biopesticide system. But we also need to address within this whole thing is, how do we detect and predict small RNAs, and how do we optimize this for making the whole process work better? So typically, we're not looking at mobile double-stranded RNA here. Typically, we spray double-stranded RNA on a leaf, and we see small RNA processing in either the pathogen or the plant, depending on what we're looking at. So small RNAs, as I said, are the key effectors of RNAi. We need to optimize these to really move forward in the field. At the moment, the methods of detecting these small RNAs from topical applications, so on the same leaf, are really not very effective. And I'll show you on the next slide what I mean by that. optimizing both the detection and production and even the prediction of these small RNAs and how we can make them better, how we can get them produced more efficiently, targeting more efficiently, or even amplifying themselves more efficiently is really key to the field moving forward, in my opinion. So what do I mean by currently detection is not very efficient? This is an example of a northern blot, but in this time it's run to detect small RNAs themselves. So small RNAs should be within here of about 21 to 24 base pairs in length. You can see a microRNA, which is a very defined band. But when we look at double-strand RNA added to a plant, either in wild type or DCL234, whole tissue analysis, we don't see any really defined bands of small RNA. And I've seen papers published where they actually said these are small RNAs. When we eliminate the other part, so if we eliminate the dices from the fungus and from the plant, and this is everything all together, we still see this smear. So this is really not a small RNA band, which unfortunately has been misinterpreted in the past as this. We've done small RNA sequencing in this in the past, so this is prior to when I got to the lab, but it basically shows the same thing. We have no real defined class of small RNA here when we sequence small RNA from a topical application, and it really reflects this non-specific smear we see here. As opposed to a normal hairpin, which is processed in plantar, this is what we expect to see. Very nice peaks of 21, 22, and 24 base pair small RNAs in a symmetrical type pattern. And you can see when we look at this on a blot, you should see 24, 21, whereas we don't see any of this here, depending on the dices we have within the system. So it really shows that the detection and prediction of small RNAs is masked by degraded double-strand RNA. So we're spraying a lot of double-strand RNA on these plants, and it really is masking what we can see and how we can really optimize the production of these small RNAs. So what's one way of looking at these small RNAs and getting rid of all the degradation products? The first is some of the things I introduced early on. So we could potentially tag argonauts. So I'll focus on fungi here. If we could tag argonauts within fungus, pull these down, irrespective of all the double-stranded RNA, non-specific small RNAs, which are in the other proteins, we can purify argonaut-bound small RNAs, and we can sequence these to generate a profile of these. So I did this previously in Arabidopsis. The possibility exists that we can do this in fungus and any pathogen we want to target. This has been proven effective, and it can resolve even argo-specifics. So I resolved previously Argonaut 1 and 2 within C. elegans. So this could, in theory, be done for fungus as well. However, it takes time to establish. It only resolves individual argonauts, so you need to double everything you need to make, because often fungus have 2, even 3 argonauts. And it requires a generation of transgenic target species. And this is either difficult to do if things are difficult to transform. Biochemistry is not so good in some of these species. And if we want to do this in any field setting, this is really not a feasible approach. So I wanted to come up with a way of purifying small RNA independently of using tagged argonauts and transgenic organisms. So lucky for me, where I previously worked, one of my colleagues was a mad biochemist, and he came up with this really simple system of generating argonaut tagging independent ways of purifying small RNAs. So we called this trans-kingdom rapid affordable purification of risk. So the RNA-induced silencing complex, which contains argonaut, and we'll call that TRAP-R from now on. So it's actually become a commercial kit. So his name was Thomas Gretzinger. And this is a commercial kit, which is now available on the market. So I'm in the middle of a seminar. Is that okay? Yeah. Sorry about that. So this is a commercial kit now, but he developed it in the lab where I previously was, and it's a way of purifying risk complex from many different organisms. How does it work? Well, he elutes under certain salt concentrations, and you can purify all of the argonauts present within a single organism. This is Arabidopsis, which actually contains 10 argonauts, and you can see he purified all of these. It removes all of the non-specific degraded or tRNAs, small RNAs, which are not actual small interfering RNAs. So you can see here, when you elute through the column, you see a really pure pool of small RNA. So it works via a Q-sepharose system. So sRNA is bound by Argonauts, actually a loop before any degraded RNAs. So it's really the perfect system that we could apply. It works in many different species. So it works in plants, C. elegans, yeast, mice. The list is basically endless. We wanted to see if we could use this in fungus. So it allows for enrichment of small RNA species when genetic modifications are not possible. So we wanted to know, can we use this to effectively enrich topical double-stranded RNA, derive small RNAs, and thereby sort of enhance the efficiency of RNAi based on this? So we first started by sequencing just botrytis. This is the gray mold you see in your strawberries at the supermarket, unfortunately. When we sequence total RNA from this, we see a relatively small peak of 21 to 22 base pair small RNA. So this is small RNAs produced endogenously in botrytis. When we put it through the TRAP-R column, we saw a significant enrichment of these 21 to 22. So this is a real enrichment of functional Argonaut-loaded small RNAs within botrytis. But where the technique really shone is when we add double-stranded RNA to this. So we added double-stranded RNA to in vitro grown botrytis cultures. And when we just sequenced total RNA, all we saw was a degradation profile. So we didn't see any enrichment in small RNAs at all. So this is reflective of the smears I showed you before on the small RNA blots, so the northern plots. However, when we put it through the TRAP-R column, we saw an enormous enrichment in 21 to 22 nucleotide. So DICER-produced Argonaut-loaded small RNAs. And this is really sort of a light bulb moment where we figured we can really resolve what small RNAs are being produced from these double-strand RNAs added, which were previously completely masked by degradation products. So this is an example of when you sequence total RNA from a plant, from a fungal culture. Basically, you see no enrichment of significant sort of symmetrical classes of small RNA. So this is really not what you should see when you see a small RNA profile. However, when we enrich this by using the TRAP-R columns, what we saw is a symmetrical profile with really enrichments in the right classes of small RNA produced by dicers loaded into Argonauts. The additional thing with this is we could also see extra species of small RNAs produced via amplification. And this is something we reason is going to be really necessary to get this technique to the next level when targeting pathogens. So TRAP-R really improves our resolution of small RNAs when we're targeting specific loci, in this case, botrytis. We also reproduce this on plantar. So these are plants treated with double-strand RNA, infected with botrytis, and then we sequence them. So again, when we added double-strand RNA, we saw degradation products, no enrichment of small RNA. But when we TRAP-R purified, we could see really nice peaks. And this again is reflected in the small RNA profiles we see. Total RNA, this is really probably the bulk of it, just degradation products. Whereas when we look at the TRAP-R purified, we see some level of symmetry here. It's still a little bit dirty, but this can really give us an indication of how to improve this in the future. So is dicer processing not accurate? Do we need to optimize the template? Do we need to optimize Argonaut loading? So what do we go beyond this? We have a ton of small RNA sequencing beyond the loci which we're actually looking at. So these are chromosome level plots done by Steve Fletcher in the lab. And you can see really interesting peaks of small RNAs. So we have an enriched set of sequencing data here for functional small RNAs. So what are these small RNAs? Do they exist in multiple fungal species? And can we utilize this to really enhance our RNAi picture within both just how RNA works in fungus and how we can use this as a protective mechanism of RNAi. So what can we learn from this? So something Steve's been doing is trying to get a machine learning up and running. So identification of genome-wide functional small RNAs to train models. So we want to really learn how to predict what small RNAs are produced from certain types of double-strand RNA. And can we train this to really optimize? So we already know we can make a double-strand RNA to target sequence specifically, whatever we want, but we want to optimize the small RNA production from this sequence. And this is something which we're actively looking at, or Steve's actively looking at. And we want to gain also new insights into the genome and transcriptional regulation within these fungus. And this is an example of that. What we can see is total RNA sequenced from botrytis. This is a unique loci near a ribosomal region. When we trap or purify this, we can see really nice peaks of small RNA, which we completely miss when we're doing input. So we really want to delve into not only within botrytis, because I've only shown you botrytis here, but we've actually sequenced from about 5 different fungal species here to optimize not only double-stranded RNA to target these fungus efficiently, but also to really get into the nuts and bolts of the RNAi machinery and try and use this to our advantage when we're trying to target them from a fundamental perspective and an applied perspective. And with that, I'd like to thank all the people who helped with this. So within the ARC Sustainable Crop Protection Hub, so Han, Steve, Don, Safira, Ritesh and Nina, the Genome Innovation Hub. So they provided all the support for the sequencing, which I showed you. So this was primarily done by Stacy and Max, and also Shiv. So she did a lot of the actual library preparation. From my time at the ETH, Thomas, who came up with the whole TRAP-R sequencing, and Manu, he was a really good collaborator in all my movement stuff. Alexi, who did a lot of the bioinformatics. And within QBI, Steve Zurin and the funders for supporting me. And thank you for attention. I'll take any questions you have.
Okay, thank you, Chris. Well, I know I personally found that a fascinating talk, even though I've seen you give talks many times now, given that you're in our group. I think that was an incredibly well-presented, comprehensive explanation, and not too complicated, hopefully, for everyone else in the audience who's not experts in RNA or follow. There are a couple of questions coming through, and I encourage everyone in the audience to please put some on. I know there are some experts in the audience as well, so please feel free to add some into the Q&A as well. I just wanted to ask you that what is it that you're going to be looking at next? So you've shown a nice progression of where you're at now. What do you see as the next phase of this research?
I think I'd really like to optimize how this double-strand RNA is processed, and can we utilize the lessons we've learned from this TRAP-R sequencing to really get an amplification process happening. So I think we're currently limited by what double-stranded RNA we add to our pathogens. And if we can utilize an amplification process, which is already present within, let's say, a fungus in this case, we can really move this to the next level where we can have a single spray, which is way more effective than having to redose it all the time. So that's one thing. The other thing is, via a different project, we're looking at seeing if we can get different versions of RNA actually into the cells of a plant. So using specific nanoparticles, but that's a story for another day.
Yeah, I think there are a lot of questions that still can be answered. It's a really interesting field to be in, I think. So I'll move on to some of the questions from the audience. So Andrew Gearing has asked about the microviral infection status of the fungus. So do you think if a fungus such as botrytis has a microvirus, would it affect the sRNA profile?
The answer is probably yes. And that's one of the things which we are trying to look at. So we have different botrytis isolates, one which likely contains a bunch of viruses like this one, like the microviruses, and another which hopefully doesn't. And we'd like to see how that specifically affects, one, the ability of the fungus itself to process small RNAs, and whether this is actually hindering or enhancing the ability to process double-stranded RNA. So yeah, that's a very good point, and something we are looking at.
Yep, thank you. And an expert question here from Peter Waterhouse. Do you think the dsRNA moves mainly through the xylem for apoplastic movement in the plant?
In general, yes, but I wouldn't put it past. There is a little bit in the phloem, and there is apoplastic unloading of double. You sound like one of the reviewers for our publication. I think it predominantly moves through the xylem, yes. There is some movement through the phloem, but in general, it's an apoplastic phenomenon, which maybe passes into the phloem to get to the roots, especially.
Yeah, so there were the publications from Ritesh showing that the phloem-feeding insects can uptake the dsRNA. So
Ritesh actually showed that it's definitely in the phloem, because whitefly specifically feed from the phloem, and he was able to knock it out. So it definitely gets into the phloem to some degree, but it's probably biased towards the xylem.
And it might be plant specific potentially, I assume.
There is differences, yes.
Okay, next question from Christoph Rosenthaler. Please apologize for your butchering your name if I have. His question is, Manfred Heinlein showed that topical application of artificial dsRNA, which is poly-IC, inhibits root growth. Do you see the same inhibition?
Not so far, although I've never done real phenotypic quantifications of this. So, yeah, that's a good question. I need to look at that phenotypically.
Okay, thank you, Christoph. And I think we've got time for a couple more questions. One more here is, hi, Chris, from Milos. Thank you, the work's very exciting. Can you speculate on the movement of dsRNA and potential amplification, especially within the dsRNA that remained intact for a couple of weeks? Do you think that they are throwing off RDR6 or similar sRNA products?
I mean, the bulk of the double-strand RNA, which we look at, never really enters plant cells. It's when it enters the fungal cell that the RDRs are actually doing some form of amplification. So if we got it into plant cells using, for instance, the nanoparticles, which, allow crossing the plant cell wall, that's distinctly possible. Within fungus, I think there is a level of amplification. So when I skipped over the GFP with a little sequence of GUS on the end of it, that was designed specifically to address transitive or secondary small RNA amplification. So what we could see there, that there was no GUS small RNAs, but there were plenty of GFP small RNAs. So these are likely amplified from the template which double-strand RNA is targeting. So it probably would happen similar in plants if it did into the cells, or if we could get it in there as efficiently as we do into the fungus.
Okay, thanks, Chris. One from Chemchung. Did you check the sRNA distribution into DCL234 after dsRNA treatment using your TRAPR sequencing?
Without fungal infection, yes, we have sequenced it. I just have to remind myself. So I don't think we saw any small RNA production. I've certainly never detected them. So, there probably is a background of them there, but we exclusively looked at in fungus mostly. So.
Okay, thank you. And a couple more questions. Is the movement from Timani, is the movement of dsRNA passive or facilitated? And if facilitated, are you looking at those factors? Are you looking at those factors?
So, I mean, we do, it's different to what you mean, I guess, but we do facilitate it by adding a penetrant. So you have to add a certain penetrant to get it past the cuticle of a leaf, and this varies between different species. After that, I believe it's largely passive, although it probably does change under, let's say, drought conditions, nutrient stresses. So, for instance, salt stress would change the ability of apoplastic flow. So in general, I'd say it kind of follows the water flow of a plant, so largely passive, but it is dependent on, those environmental factors.
Okay, thanks, Chris. We've got another 10 minutes, so a couple more questions. Rakesh David has, hi Chris, great talk. Have you looked at RNA secondary structure as a means of improving the RNA movement efficacy? So
I personally haven't.
Sequences have been.
So I personally haven't, but you can probably talk to Cheng Cheng about this, because that's kind of what she's doing within Bernie's lab. So Bernie looks at unique TLS structures based on Fritz Kraegler, so one of Bernie's big collaborators. So he showed that mRNAs actually move across graft junctions. And once he showed that, he looked into the structures of these RNAs, and he found unique structures of TLS-like, so structures on the ends of these RNAs. And Bernie's actively looking, and Cheng Cheng is actively looking at adding these to RNAs to see if they enhance movement throughout the plant. So if you are really curious, I would ask Cheng Cheng or Bernie, because they've got way more knowledge in this than me.
Okay, I think that's probably the last question that we've got from the audience. So we might wrap up for today. So thank you everyone for attending our seminar. Please visit the Coffee Science Seminars webpage for more information on our upcoming seminars. And if you haven't already signed up to the invitation list, I think there's maybe a QR code on the next slide, Chris, is there?
Yes, there is.
Okay, if you haven't already signed up for the seminar, please do so at coffee.uq.edu.au/science seminars, or use the QR code at the end of this seminar, and you can just check the recording. And I think we'll wrap up here. So thank you everyone for joining us, and thank you, Chris, for a really engaging and interesting talk for us today. And we'll see you at the next seminar, all the audience members. Thank you.